This presentation is part 2 of 3 of the digestive system. We're beginning this slide presentation with the mouth, which is where ingestion begins. And encircling the mouth, we see the lips or labia. There is a frenulum, a labial frenulum, on the medial aspect of the upper and lower lips that joins the inner portion of the lip to the gum. The labia are highly vascular and they are also innervated. The oral vestibule or oral orifice is that anterior opening, and where the pharynx begins is what's known as the fauces. That's the posterior opening of the mouth where the pharynx begins. On this picture you can also see the hard palate and the soft palate. The hard palate is where we have the mucous membrane covering the palatine bone as well as the palatine process of the maxilla. And then the soft palate is that continuation of mucous membrane which ends in an inferior portion known as the uvula. So here we can see the uvula, and remember the uvula is going to help close off the nasopharynx from the oropharynx during swallowing. So the soft palate is actually made up of skeletal muscle. Again, the soft palate will rise to close off the nasopharynx when we swallow, and the soft palate is anchored to the tongue laterally and posteriorly. The tongue is important because it helps with ingestion, mechanical digestion, chemical digestion, sensation, swallowing, and vocalization. There are papillae, which are nipple-like projections found on the surface of the tongue, and the fungiform papillae, which are scattered all over the surface of the tongue. Those are those little dots that you see. Those house taste buds. There are also filiform papillae, which kind of give the tongue its whitish appearance, which help make the tongue a little bit rougher for licking. And we also have foliate papillae on the side and valate papillae in the posterior aspect of the tongue. The tongue is a muscular organ. It has intrinsic skeletal muscle fibers that are oriented in different planes to allow the tongue to change shape for speech and for swallowing. The extrinsic muscles of the tongue extend from the origins of the skull and soft palate to allow for motion, so we can push the tongue laterally, we can protract it, and retract it. Within the mouth, we have 3 major salivary glands that will help to mix the food that we ingest with saliva that they produce to form a bolus that will be swallowed through the pharynx, travel down the esophagus into the stomach where it mixes with hydrochloric acid to form chyme. Now, the 3 major salivary glands include the parotid salivary glands, which are located about here, the submandibular salivary glands, and the sublingual salivary glands, which are underneath the tongue. Saliva is approximately 95.5% water. It also includes ions and glycoproteins and enzymes and growth factors and waste products. Saliva contains amylase to help the chemical digestion of carbohydrates. It does help to lubricate, and it does contain chemical buffers, including bicarbonate and phosphate and protein buffers. It includes the immunoglobulin IgA and is regulated in terms of the production by the autonomic nervous system in terms of production of saliva. And saliva is produced by 2 types of cells, including serous cells and mucus cells. Now, there is lingual lipase that is a part of saliva. It contributes in a very minor way to the chemical breakdown of lipids, but it doesn't really play a major role. I have included it in the past, and I just wanted to make sure I made mention of it again. As far as the teeth go, we have 20 deciduous teeth with which we are develop as children, and 32 permanent teeth. The 32 permanent teeth include 8 incisors, 4 cuspids, 8 premolars or bicuspids, and 12 molars. The gums are referred to as gingivae. Each tooth consists of an upper portion that you can see above the gum line that is referred to as the crown. And deep to the gum line we find the root. Unlike bones, enamel does not contain any living tissue, and enamel is found on the surface of the crown of the tooth. And deep to the enamel we have dentin. The pharynx is a fancy term for the throat, but it is broken down into 3 portions. We have an area of the pharynx that is designed for respiration solely, and that is the nasopharynx. If you look at this picture, you'll notice that there is an opening in the nasopharynx. I'm going to try and circle it here for you. I know it's the same color. But that opening is where the eustachian tube or auditory tube connects with the pharynx, and there's a flap over that to help equalize pressure for the tympanic membrane. So we've got the nasopharynx, the oropharynx, which has respiration and digestion passageways. The oropharynx is posterior to the oral cavity and inferior to the nasal cavity and posterior to the nasal cavity. The oropharynx is superior to the larynx, which is where we find the laryngopharynx. So again, the pharynx consists of the nasopharynx, oropharynx, and laryngopharynx. Please take note that this word is pharynx, not pharynx, and the larynx is not the larynx. We'd have to have the N in a different spot for those words to exist, and they don't. We have pharynx and larynx. The esophagus is a muscular tube that connects the pharynx to the stomach, and this tube is normally in a collapsed state unless it's engaged with the passage of food or liquid. The passage of food or liquid is actually regulated by 2 sphincters. We have upper and lower esophageal sphincters. Remember, sphincter muscles are just circular muscles that can close off areas to regulate the passage of different items through those areas. The esophagus, like the mouth and the pharynx, is lined with stratified squamous epithelium that helps protect against erosion. Now, sometimes if there's acid that refluxes from the stomach into the esophagus, that can start to change the stratified squamous epithelium and replace it with simple columnar epithelium. And that's either a condition known as GERD with gastroesophageal reflux. Sometimes this can happen with esophageal adenocarcinoma or Barrett's esophagus. So if the stratified squamous epithelium starts to change into simple columnar epithelium where it's not supposed to do that, that's generally considered a problem. The stomach has 4 main regions, including the cardia, which is where the esophagus enters into the stomach, the fundus, which is that upper curved portion of the stomach, the body, which is the main portion, and the pylorus, which is the lower portion of the stomach, which is where we find the pyloric antrum. An antrum is a nearly but not completely closed chamber, and this is where we find the pyloric canal and pyloric sphincter. The pyloric sphincter as all sphincter muscles are circular muscles which regulate the flow of contents from one area to another, the pyloric sphincter regulates the passage of chyme into the duodenum. The stomach contains the same 4 layers as the rest of the alimentary canal. However, it is modified. There is a 3rd layer of the muscularis externa, which the innermost layer includes the oblique muscle layer. The internal portion of the stomach, the inner lining of the stomach, contains gastric pits, which go inward to form gastric glands, where we find parietal cells, which not only produce hydrochloric acid, but they also produce intrinsic factor for the absorption of B12. We also find chief cells, which are responsible for producing the protein pepsinogen, which is the precursor molecule to pepsin. So pepsinogen actually gets cleaved into pepsin in the presence of hydrochloric acid. We've taken a look at the main hormone produced and secreted by the stomach, which is gastrin. Next, we're going to take a look at some of the other hormones that the stomach secretes. The first of which is ghrelin. Ghrelin is produced by cells in the internal area of the stomach where we find the fundus, that's that uppermost smooth curved portion. And during a fasting state, when a person is not eating, the area of the stomach, known as the fundus internally, will produce and secrete ghrelin, which acts on the hypothalamus. That will stimulate hunger, and if a person has eaten a lot of food where it's actually reaching the fundus internally, that can also stimulate the feeling of satiety or feeling of fullness. Histamine is a hormone that can be secreted by the stomach mucosa in the presence of food. I've heard previously that the act of eating is an inflammatory process, and this makes sense when we see that histamine is produced and secreted by the stomach mucosa in the presence of food. That actually acts on the parietal cells in the stomach to produce and release more hydrochloric acid. Serotonin is a hormone produced and secreted by the stomach mucosa in the presence of food to act on the stomach muscles to cause contraction. And gastric inhibitory peptide hormone is a hormone produced by the stomach mucosa and the duodenum that, in the presence of food and sympathetic stimulation, will cause the stomach, the pancreas, and the small intestines to restrict their secretion and slow down motility, slow down emptying, and slow down absorption. There are 3 phases of gastric secretion. When we talk about gastric secretion, we're focused on the secretions just within the stomach that are going to further chemical digestion of the bolus as it mixes with hydrochloric acid to become chyme, as well as the enzymes that are produced and secreted to further the hydrolysis of proteins. The 3 phases of gastric secretion include the cephalic phase, remember cephalic means head, the gastric phase, and now we're focused on the stomach, and then the intestinal phase. The cephalic phase begins with the sight, the smell, and the thought of food. Those stimuli are going to stimulate the cerebral cortex, which will send signals to the hypothalamus and the medulla oblongata. Stimulation of taste and smell receptors can also stimulate the hypothalamus and medulla oblongata to stimulate the vagus nerve, which helps to prepare the stomach by producing and secreting more hydrochloric acid and enzymes. When we enter the gastric phase, that lasts for approximately 3 to 4 hours, and as the stomach distends or stretches, which is going to occur when the bolus enters the stomach from the esophagus, that will further stimulate vasovagal responses, which again stimulate the medulla oblongata and the vagus nerve, and will stimulate local reflexes to continue peristaltic responses of muscular contraction as the presence of food chemicals, especially peptides, caffeine, and rising levels of pH are going to activate chemoreceptors within the lining of the stomach to stimulate G cells to produce gastrin, which, as it's released into the blood, will stimulate the cells within the stomach and the parietal cells to produce and secrete more hydrochloric acid and produce more pepsinogen, which gets cleaved into pepsin to help break down proteins in the stomach. And that will mix together with all of that hydrochloric acid as peristaltic contractions start to move the acidic chyme through the pyloric sphincter from the stomach into the duodenum, which is the first part of the small intestines. We then enter the intestinal phase of gastric secretion, and it's the presence of that very acidic chyme, which has a low pH, the presence of partially digested foods, fats, and that hypertonic solution in the duodenum that is going to stimulate the intestines, again, the duodenum specifically, to briefly release intestinal gastrin, which makes that stomach secrete a little bit more hydrochloric acid. Now, those are all the stimulatory events that can cause the release of gastric juice. However, there are inhibitory events that can occur, and if a person is experiencing depression or a loss of appetite, which is not uncommon when a person is feverish, there's a state of anorexia, which is just a loss of appetite that can occur when a person is feverish. It's different from anorexia nervosa, which is that eating disorder. But the loss of appetite or depression will inhibit the cerebral cortex, which inhibits the stimulatory impulses that would be normally coming from the parasympathetic centers of the medulla oblongata. So now we have inhibition of the vagus nerve, and there tends to be a buildup of excessive acid in the stomach, because the stomach will normally go through phases throughout the day of preparing itself to receive food by increasing its levels of acidity. So as the stomach becomes more acid, that actually will inhibit G cells, because if we've got enough acid in the stomach, we don't need G cells to produce more gastrin, so it inhibits the G cells, and gastrin secretion will decline. And if a person is emotionally upset, that's going to stimulate the sympathetic nervous system, which overrides the parasympathetic controls. So we already know that when we have sympathetic nervous stimulation activated, we have decreased activity in the digestive tract. And so as the duodenum will start to descend with the presence of fatty and acidic and hypertonic chyme or irritants in the duodenum, because these things will move through from the stomach to the duodenum, that actually inhibits local reflexes. It inhibits the vagal nuclei in the medulla oblongata and inhibits the pyloric sphincter, which actually will stimulate an enterogastric reflex, which inhibits the secretory activity of the stomach. And again, the distension and presence of fatty, acidic, partially digested foods in the duodenum will inhibit the release of intestinal hormones. So we now have the inhibition of secretin, cholecystokinin, and vasoactive intestinal peptides. So there can be stimulatory and inhibitory events that will affect the release of gastric juice. However, in normal circumstances, 3 phases of gastric secretion to produce gastric juice. We've got the cephalic phase, the gastric phase, and the intestinal phase, where even though once we go from the stomach to the duodenum, we are going to slow down what's going on in the stomach. Those are the 3 phases, and the intestinal phase does have both excitatory and inhibitory factors that are regulated by the duodenum. Within the stomach, the mucosal barrier is really important because it is exposed to highly corrosive, low pH gastric juice. That hydrochloric acid could do a lot of damage were it not for the mucosal barrier. So the stomach is protected from self-digestion by that mucosal barrier, and there is a thick coat of bicarbonate-rich mucus which protects the epithelial cells. The epithelial cells are joined together by tight junctions. So in between the cells, if this hand is a cell and this hand is a cell, in between them there are tight junctions that kind of hold those cells together so stuff can't leak through them. And stem cells rapidly replace the epithelial cells, the simple columnar epithelium, to help make sure that things don't leak through the simple columnar epithelium, and those surface cells are replaced every 3 to 6 days. Now, sometimes homeostatic imbalances can occur, and an ulcer is a great example of a homeostatic imbalance where acid can break through the mucosal barrier and start to eat away at the mucosa underlying the barrier. And so normally mucosal secretion is inhibited by bacteria. So stomach ulcers are usually due to an overgrowth of certain types of bacteria that eat away at the mucosal barrier. Another thing to understand is that if a person is ingesting a lot of nonsteroidal anti-inflammatories, Advil is a great example of that, that can also cause the decreased production of prostaglandin E. Prostaglandin E is a chemical produced by the epithelial cells that normally helps protect the mucosal barrier. So if a person is ingesting a lot of nonsteroidal anti-inflammatories, that inhibits the production of prostaglandin E, and that can also put the mucosal barrier at risk. They found out that when nonsteroidals were initially being taken by people who were experiencing arthritis, a lot of elderly people started taking these nonsteroidal anti-inflammatories. They were going to the hospital because they were hemorrhaging internally because of the ulcerations that they were experiencing that they didn't even know they had because of these nonsteroidal anti-inflammatory drugs inhibiting the production of prostaglandin E. This takes us to the digestive functions of the stomach. There are both mechanical and chemical mechanisms that are involved in food digestion in the stomach. The mechanical functions include peristaltic contractions, which occur about every 20 seconds, and they occur as a ripple from the fundus to the pylorus, and that helps mix the bolus with acidic gastric juice to turn it into what we call chyme. Small amounts of chyme are then going to be squirted through the pyloric sphincter into the duodenum to neutralize stomach acid and digest nutrients. A typical meal takes about 2 to 4 hours to be emptied from the stomach into the intestines. The chemical functions of the stomach include the levels of acidity, which help to break down the bolus, and salivary amylase and lingual lipase will also be included in the actions of breaking down the bolus chemically. We also have pepsin and hydrochloric acid, which begin breaking down proteins into peptide chains. During infancy, we have a special enzyme called renin, which helps digest milk proteins, and intrinsic factor is also produced by the cells in the stomach that will allow the absorption of vitamin B12. B12 is really important when it comes to producing erythrocytes, and it's also important for proper neurological function. Pepsin is an enzyme that is produced in the stomach, and this is done because chief cells will secrete an inactive protein that's referred to as a zymogen called pepsinogen. When pepsinogen makes contact with hydrochloric acid, the hydrochloric acid cleaves off or cuts off several amino acids, and the pepsinogen is turned into pepsin, which is the active enzyme that helps to further break down proteins within the stomach. In this particular picture, we can see the gastric pit that dives down, becoming a gastric gland. Let's take a look at how the parietal cells produce and secrete hydrochloric acid. You'll notice in this picture at the top, there's a picture of a gastric gland, and the parietal cells are in blue and the chief cells are in purple. And if we look at the lower portion of the picture, you'll notice that there's a blood capillary to the far left. Carbon dioxide will diffuse through the capillary wall because we know carbon dioxide is lipid soluble. It passes right through plasma membranes, and as it diffuses from the blood capillary through the interstitial fluid, it will enter the parietal cells, where when carbon dioxide mixes with water in the parietal cells, with the help of carbonic anhydrase, it will turn into carbonic acid, that's the H2CO3, which will easily and readily dissociate into a molecule of bicarbonate. That's the molecule that you see below the H2CO3. Here's the bicarbonate. And it will also form a free ion of hydrogen. So the carbonic acid dissociates, and then the carbonic acid, as it's associated into bicarbonate, what's going to happen is that we're going to follow the bicarbonate for just a second. The bicarbonate will exit that parietal cell. Remember that when bicarbonate exits a cell, we're going to have chloride move inward as the bicarbonate moves outward. That shift of bicarbonate out of the cell is also referred to as the alkaline tide. You can see that term there. And again, that exchange of bicarbonate and chloride is antiport, where we've got ions or molecules moving in opposite directions. So now we've got chloride inside the parietal cell, and there's a protein channel that allows the chloride to move through the parietal cell into the lumen of the stomach. So here we can see that the chloride has come out here. Now let's go back up to the top of the picture where we have that hydrogen ion from the carbonic acid that dissociated. That hydrogen ion is going to move outward from that parietal cell into the lumen of the stomach. Here we can see it's moving outward as potassium moves inward. So that's also antiport because it's moving in opposite directions. And now that we have hydrogen in the stomach lumen and chloride in the lumen, remember those will form an ionic bond of hydrochloric acid. And the hydrogen and the potassium need ATP in the form of hydrogen potassium ATPase. Okay, that's the enzyme pump that's going to drive the hydrogen into the stomach lumen while the potassium goes into the parietal cell. So hydrochloric acid is going to build up inside the stomach as bicarbonate builds up in the blood. And again, that's what we refer to as the alkaline tide when the extracellular fluids, before that bicarbonate goes into the bloodstream, the extracellular fluids become more alkaline when digestion occurs in the stomach. Let's take a closer look at feedback control of gastrin secretion. When ingested food arrives into the stomach, it actually buffers the stomach acid, so we have a rise in pH. And the elevated levels of pH are going to stimulate the G cells. And as the G cells are stimulated, we see those in kind of a darker pinkish purplish color. When the G cells are stimulated, they're going to secrete gastrin, as we know. The gastrin stimulates sheath cells and parietal cells, and the parietal cells will secrete hydrochloric acid, while the sheath cells are going to secrete pepsinogen. That pepsinogen, which is an inactive enzyme or zymogen, is going to be converted into pepsin by the hydrochloric acid. We're over here now. And so now we have active pepsin. The pepsin helps to digest dietary proteins, and then those dietary proteins, as they're partially digested, they become groups or clumps of scanty chains of peptides. Oligo means scanty, so oligopeptides just means it's a brief chain of amino acids. And so those oligopeptides and other amino acids, free amino acids, are going to further buffer the stomach acid. When we get into acid-base balance, proteins can act as buffers, so they're going to soak up excess hydrogen ions, and so they're going to elevate the level of pH. And so we're really getting a positive feedback loop here. And we can see that oligopeptides can also directly stimulate the G cells. So that's a positive feedback control mechanism for gastric secretion. There are a lot of hormones involved in digestion, including one we haven't mentioned before, which is motilin. Motilin is a hormone that's produced by the mucosa in the duodenum, and it's stimulated to be produced during fasting. And there's a periodic release of this hormone that occurs every 1.5 to 2 hours by neuronal stimulation in the duodenum, and it stimulates the proximal duodenum to begin a series of muscular contractions known as the migrating motor complex. And these muscle contractions actually occur in the stomach and the small intestines during fasting to act kind of like the housekeeper for the gut. Its purpose is to help clear undigested food particles and bacteria and debris from the small intestines and help move them to the colon. And that also helps prevent conditions like small intestinal bacterial overgrowth, known as SIBO, S-I-B-O. And there are different factors that can affect the migrating motor complex. Eating interrupts that pattern and that cycle, stopping contractions until the next fasting period occurs. Parkinson's disease and certain gut infections can impair this process. Sometimes antibiotics can stimulate it. So erythromycin is one of those types of antibiotics that can actually stimulate the migrating motor complex. We've already discussed secretin, and I believe we've discussed serotonin, but I'll say it again. It's produced by the stomach mucosa, stimulated by food in the stomach, and it causes contraction of the stomach muscles. Somatostatin is a very inhibitory hormone. It inhibits gastric secretion, it inhibits secretin, it inhibits secretion of the pancreas, it inhibits gastrointestinal blood flow, and therefore inhibiting the absorption in the small intestines. It inhibits contraction and bile release from the gallbladder and liver. There's another hormone known as vasoactive intestinal peptide hormone, and this is a hormone that is produced by enteric neurons. Those are the neurons in the submucosal layer of the alimentary canal in the small intestines, and it's also produced by the pancreas and stomach. It stimulates buffer secretion. It helps to dilate intestinal capillaries, relax intestinal smooth muscle. It increases secretion by the pancreas and inhibits acid secretion by the stomach. We also have cholecystokinin, which we did discuss earlier. We have gastroinhibitory peptide hormone, which is also sometimes known as glucose-dependent insulinotropic peptide, and that's produced and secreted by the mucosal layer in the duodenum. It's stimulated by the presence of fatty chyme. It will act on the stomach to inhibit production of hydrochloric acid to a small degree, and it does stimulate the release of insulin by the beta cells in the islets of Langerhans in the pancreas. We did talk about gastrin, we did talk about histamine, and intestinal gastrin is something that we did mention that's produced by the mucosal layer in the duodenum that will stimulate gastric glands and motility in the stomach. This is going to take us to vomiting. What a way to end on this particular video presentation, part 2 of 3. So vomiting is the forceful ejection of stomach contents, and it's a muscular action that's coordinated by a special group of nuclei in the medulla oblongata known as the emetic center. And it's caused by lots of different things. Sometimes overdistension or stretching of the stomach can cause this reflex, or overdistension of the duodenum can also cause that reflex to occur. Alcohol, bacterial toxins, visceral trauma, intense pain, repugnant thoughts, smells, and sights can cause this reflex to occur. Vomiting is often preceded by retching or profuse salivation. If a person starts salivating profusely all of a sudden and feels really sick, that's not a good sign. The retching can also cause pressure differences, which open up the sphincter muscles and dilates the esophagus. Vomiting can also cause tachycardia and sweating. The process of vomiting and getting rid of the stomach acid, the hydrochloric acid, can lead to temporary alkalosis if a person keeps losing acid contents from the stomach. So that's important to know. That's going to be the last slide for this presentation. We're going to move on to part 3 of 3.